Okay, so what we're looking at today is just the afferent division of the nervous system. So we talked about the afferent and the efferent divisions of the nervous system last week. One of them is sensory and one is motor. So the afferent division, does anyone remember, is it sensory or motor?
Afferent sensory.
Sensory, yes, exactly. So we're going to be looking at the sensory side of the nervous system today. So the afferent division of the nervous system is made up of sensory receptors and sensory pathways. The sensory receptors, we will take a look at the different types of sensory receptors that we have. All different types of receptors give us different bits of information. So all the sensations that we feel come from the different sensory receptors. So we're able to feel temperature. We have receptors specifically for temperature. We're able to feel pain. We have receptors specifically for pain, and so on and so forth. The receptors receive their little bits of information, and they send those bits of information along the sensory pathway and deliver that information to the central nervous system, the brain and the spinal cord. And remember that we can divide the afferent division into two subcategories. We have somatic and visceral. So the somatic sensory information is information that we receive from the exterior environment. So someone tapping you on your shoulder, something pressing up against your skin, that is external sensory information. That information is sent to the primary sensory cortex or areas of the cerebellum, just depending on what the particular bit of information is. Then we also have visceral sensory information. Visceral sensory neurons give us information about what's going on internally. We will look at specific examples today, but a good example is how you can feel when your bladder is full. You feel that sensation of fullness. It comes from stretching of the bladder. So that information is sent along the visceral sensory pathways to reflex centers in the brainstem or the diencephalon. Also, just depending on what the specific bit of information is. Is that rain? Is that sound of rain?
I think it might be a fan on my laptop.
Oh, darn. Got me all excited. I love the rain so much. Okay, sorry. Total aside there. So, for our senses, we can put all of our senses into two big categories. We have general senses and special senses. Special senses we will take a look at the end of the semester in its own special section because they work a little different. They're special. Today, what we will look at are all general senses. General senses is stuff like temperature, pain, pressure, touch, vibrations, and then things that you can feel internally as well. These are all general senses. General senses are general senses because there are sensory receptors for your general senses that are spread all throughout the body. For example, if I were to take a pin and poke you on your arm, you could feel that on your arm. And you could feel it on your leg and your head, and you could feel it basically anywhere I poke you. It's because it's a general sense. So we've got receptors for those all over the body. Whereas with special senses, you can only receive taste information on your taste buds, right? You can't taste with your elbow, right? You can only receive visual information with the sensory receptors in your eyes that are for specifically light. So that's really the difference. For general senses, we receive a sensation. That is the actual bit of information that arrives at the receptors. Then that information travels along the sensory pathway to our central nervous system, brain, and then we perceive that sensation. So perception is being consciously aware of that sensation. And this is where we start to see individual differences come into play. You could create a poking machine that pokes people in the exact same spot with the exact same hardness, and then you know you're delivering the same sensation to each person, but their perception of that sensation is going to be different based on how the brain interprets that information. The higher the perception is basically a difference of how many action potentials and the frequency of action potentials that we're receiving. So when someone taps you very lightly on the shoulder, that's firing off some action potentials, say relatively slow, versus someone really pats you on the shoulder like this, and we have a much higher frequency of action potentials. And that's the difference between perceiving a light touch versus a really hard tap. Okay, our sensory receptors are able to adapt to the sensations that are coming in. And there are different levels of adaptation based on the different types of receptors. And also, we have two different types of adaptation. So you can have peripheral adaptation and central adaptation. Peripheral adaptation is where the receptor activity changes. Meaning, the receptor itself can adapt so quickly that the sensations that are coming in once adaptation occurs are not perceived as intense. So fast adapting receptors, as an example, we have thermoreceptors. Thermoreceptors are our sensory receptors for temperature. So this room, as an example, if this room were to slowly get just a little bit warmer, just bit by bit, our thermoreceptors would adapt to that change in temperature pretty quickly. To the point where at the end of our time in class, we might not even really be consciously aware that that adaptation has occurred. So we wouldn't be consciously aware that the room is warmer. But when we leave the room and we go outside and it's suddenly much colder, that big change we would be aware of. But small changes, not so much, because our thermoreceptors adapt very quickly. Compared to slow adapting receptors called nociceptors, these are our pain receptors. Pain receptors are slow adapting. This is basically there for our protection. We would not want fast adapting pain receptors because pain is the signal to ourselves that says, don't do that thing. That thing is going to hurt you more. It's going to cause more tissue damage. So this is why, and I think I've asked people before, I know, I hear the rain too. It makes me so happy. I think I've mentioned in class before, rolling an ankle. We talked about that. Like probably everyone's rolled an ankle at some point. And have you ever felt like casually throughout the day you step kind of weird and you don't actually roll your ankle, but you almost do it, and it makes you really like, oh my gosh. And you can think of that time that you did roll your ankle really bad, and it almost hurts again just for a second. That's because our pain receptors, our nociceptors, are so slow adapting. It can remind us of an old injury just by doing something kind of similar. And again, that's there for protection. So that pain signal is, oh, remember that one time you rolled your ankle really bad? Well, don't do that thing again. Don't step like that because we don't want to hurt ourselves. So again, that's peripheral adaptation where the receptor activity changes. Different from central adaptation. So when we receive the sensory information, it's received at the receptor. And then that sensory information travels along the sensory pathway until it gets to the central nervous system. Central adaptation is where the nuclei along the sensory pathway become inhibited. So that sensation is still received, but as it makes its way to the central nervous system, the perception of it goes down. By the time it reaches the brain, we're much less aware of it, or even not aware of it at all. So a good example of this one would be how a new smell after a certain amount of time just disappears. So think of a time that you have been to somebody else's house, and you walk in and it very obviously smells like somebody else's house, right? Like their laundry smells different, or maybe they're cooking some food and it's really different from your own house. And it's just like, wow, I'm in someone else's house. This smells very different. And then after a few minutes, you're not thinking about that anymore at all. That smell basically goes away. Well, all of those smells are still there in the house. So all of those smells are still hitting the sensory receptors in the nose, but along the way to the brain, the nuclei are being inhibited so that you are no longer consciously aware of that sensation, and that's adaptation. So our general senses we can break into main categories. Exteroreceptors give us information about the external environment. Proprioceptors, we talked about before, give us information about the position of our body. And interoreceptors give us information about what's going on inside. These are just general categories, and then we'll look at the specific receptors that fit in those categories. Okay, so nociceptors give us information about pain, thermoreceptors, temperature, mechanoreceptors is anything that physically distorts the body. So all different variations of like touch or vibration would fall into that category. And then chemoreceptors give us information about chemical concentration, and that's happening inside the body. Yes? No? Okay. So we'll start off with nociceptors, pain receptors. We have pain receptors sprinkled all throughout the body. We know this. We can feel pain wherever. Most of these pain receptors are located in more superficial layers of the body. So all throughout the skin, within joint capsules. We do have them within bone, which is why breaking the bone hurts. We do have pain receptors within the walls of blood vessels, which is why we can feel pain when we're bleeding. But when we go deeper into deep tissue and visceral, like internal organs, we don't have a lot of nociceptors, not a lot of pain receptors. So they're more so on superficial layers. These pain receptors are called free nerve endings, and I have a picture coming up, but it's basically kind of branched out like this, and it can have a wide area in which it receives information, which would give it a large receptive field. So one receptor, one nociceptor, can give information across like a wide area. It just depends. Because it has a large receptive field, it can make it hard to pinpoint exactly where the pain is coming from. We'll look at some specific examples. These nociceptors give us information about extreme temperature changes. So not the same thing as our thermoreceptors, which can tell us when we're going from like a nice cozy warm room to a cold outside area, but extreme temperature changes, like you're for some reason grabbing onto a brick of ice and it starts to really burn, or you touch a flame and that also burns. Mechanical damage would be something jamming into you, running over you, being pushed really hard, and anything like that, large and forceful, is mechanical damage.
Or a cut.
Or a cut, yeah, right, or a cut. Dissolved chemicals, this would be something that happens internally when cells have been injured and they release chemicals, so then we're aware of cell injury and cell death. And when we sustain an injury, all three of these sensations can be triggered at the same time, which is why sometimes we describe pain as like a burning sensation. Oh yeah, so here we go. Here's a nice visual of what the free nerve ending looks like. So here we have two noisy receptors, and this would be the receptive field. And you can see how the receptive fields are relatively large, and it's just because the free nerve endings are kind of spread out. Yeah?
Do they overlap in any instances, or usually not really?
It depends on where it would be located in the body. Actually, I'm going to expand on this just a little bit because we have different types of nociceptors. So let me go through this, and I think I might answer your question. So there's different types of nociceptors. We have type A fibers. Type A fibers are myelinated. So last week we talked about the difference between a myelinated axon and an unmyelinated axon. Remember, the axon is just the name of that highway of the neuron, of the nerve cell, that the action potential travels down. So an axon can be myelinated, meaning it has a wrapping around it, and that wrapping allows for that signal to be sent very fast. Or an axon can be unmyelinated, and it does not have the wrapping, in which case the signal will be sent slower. So these type A nociceptors that are myelinated they give us information quickly about fast pain. So an example for this would be like when you get an injection, like a shot, you can feel that sharp pain feeling. Or when you get cut or hit really hard. So that type of pain sensation, we are able to pinpoint to more of a specific area. If we get cut, well, one, we're able to see it, so you can look at the area, but you're able to pinpoint more specifically, like, yeah, it hurts like right exactly here. Versus when we trigger type C fibers, which are not myelinated, this is more of the slow pain. This is not as specific as an area. This is more of like, oh, I hurt my back. Oh, my back hurts. Oh, yeah, wow, where's your back hurt? Well, it's like kind of this area over, it's like between here and here. And you can't say exactly where. You couldn't point to like, right exactly here is the spot. It's more of a general area, and that's type C fibers because it's more of a larger receptive field and general location. Now, pain perception is interesting because we all experience pain differently. We all have these nociceptors. We all have the type A and the type C fibers, where we all receive that information from these sensory receptors, from these slow adapting receptors. These sensory receptors are going to continue to respond to that pain stimulus as long as that stimulus is still present. So if you get hit, ouch, and you keep on getting hit, that sensory receptor is going to keep on working and keep on delivering that information because it's slow adapting. Again, this is for our protection. We need to know this information because if we just stand there and keep getting hit and we're having tissue damage, that's not good for the body. So the pain sensation says, hey, like, move away if you can. But here's where we start to see individual differences, is this stimulus, this sensation, is facilitated by neurotransmitters. Facilitated, in this case, means help work stronger. So remember that neurotransmitters are chemicals that are released by neurons when they communicate. So if person A releases a lot of neurotransmitter, when they are feeling pain sensation, and person B doesn't release as much neurotransmitter, they're going to feel that pain differently. So depending on how much neurotransmitter is released, that pain sensation is going to be different. On the other hand, you can also have that pain sensation decreased by endorphins and something similar to an endorphin, it acts in a similar way called pancreathalin. And this can change the perception of pain. So where neurotransmitters can increase how much pain you're feeling, things like endorphins can decrease how much pain you're feeling. So a person that doesn't release a bunch of neurotransmitter and also happens to release a bunch of endorphins is not going to perceive pain the same way as someone who releases a bunch of neurotransmitter and doesn't release a lot of endorphins. Yes?
So, like, when you run a long distance, your muscles hurt, but you don't necessarily notice it until you stop because you're getting that runner's high.
Yeah, absolutely. And with that specific example, then we start to look at things that we talked about in the muscles lecture when it comes to muscle fatigue and the psychological effects. And for questions like that, there's usually more than just one factor that comes into play. But yes, absolutely. If you're doing an activity that is releasing endorphins, like you're running, for example, and maybe there was something at the bottom of your shoe, and it's kind of rubbing on you a little bit, but you're releasing endorphins at the same time, so you're not really perceiving that pain very much. Yeah, that would be a great example. But isn't that interesting? Because everyone has different thresholds of pain tolerance, right? So this is why. Okay, thermoreceptors give us information about temperature. These are also free nerve endings, so structurally they look the same as those noisy receptors. And we have these also sprinkled throughout the skin. We also have them in our skeletal muscle, in our liver, and the hypothalamus. Hypothalamus is a region in the brain within the diencephalon. And we have about 4 times more cold receptors than warm receptors. And these, of course, are sensitive to when temperature changes, but they adapt very quickly, which is why we are able to perceive big changes in temperature much more easily than small changes in temperature. Okay, mechanoreceptors. This is a category. Mechanoreceptors give us information about physical distortion, but there's lots of different types of physical distortion, and we have different receptors for each different type. So tactile receptors give us information about touch. If you were to touch something with your fingertips, you could feel it, or pressure that's placed upon you, or vibrations. Those are all tactile receptors. Baroreceptors, like the word barometer, baroreceptors measure changes in pressure. We have baroreceptors internally. So baroreceptors within the walls of our blood vessels, within the bladder, that give us information about how full our bladder is.
Inner ear.
Inner ear, baroreceptors in our inner ear.
Like pop your ears, right?
Yes. We have special receptors in our inner ear because our inner ear is actually a special sense organ. It's probably more detailed than you were thinking. We'll talk a whole bunch about the inner ear. There are extra specialized cells in the inner ear. But yes, we can feel that pressure, but that's probably not from baroreceptors. It's probably from something else. Our digestive tract, like you can feel when you're very, very full. You can feel when you have to go to the bathroom. And then proprioceptors is the position of our body, our joints and muscles. So we'll talk a little bit about the different types of mechanoreceptors. So tactile receptors first. We can break tactile receptors down into two main categories. We have receptors that give us information about fine touch and pressure. This is detailed information. We're going to find these types of fine pressure and touch receptors in areas that are really, really sensitive. They also have very narrow receptive fields, so we're able to pinpoint very closely where that information is coming from. And the other category would be crude touch and pressure. These have larger receptive fields, and we're not able to get as much detailed information. So here are some examples of some tactile receptors. So one is just a regular free nerve ending, which we can see up here. It looks just like the noisy receptor, and it looks just like the pain receptor. So structurally, it's the same design, but this is a tactile receptor. It sits between the epidermal cells, so this is the skin. We also have some on the surface of the eye, and this gives us information about touch and pressure. A lot of these tactile receptors, actually all of these tactile receptors, will give us information about touch and pressure, but some will be fine touch. It's a slight difference there. This one down here is a root hair plexus. So you can see here is the hair shaft. This might look a little familiar. Here's the hair shaft. Here's the erector pili muscle that gives us goosebumps. Here's the hair follicle at the bottom, and then wrapped around the hair follicle, we have this root hair plexus. When hairs move, that moves the hair, and that is going to move the root hair plexus. That triggers the action potentials to be sent from the root hair plexus, travels along the sensory pathway, and then tells us something's moving your hair. Merkel's discs, I don't have a picture up here. This is fine touch and pressure. And we see these deeper within the epidermis and the stratum basal. Then we have Messenger's corpuscles, also give us information about fine touch and pressure. These are fast adapting, so we see them in areas that are very sensitive. So eyelids, lips, fingertips, and then other sensitive areas, nipples, external genitalia, Messenger's corpuscles. And then Pacinian corpuscles, these give us information about deeper pressure. So they're not so much on the surface, not on the epidermis, they're located on the dermis. And then some internally within the intestines, the pancreas, and the urinary system. I have a picture here. So this would be an example of just a chunk of skin, just any regular chunk of skin. And we can see that we have a mix of all these different sensory receptors. And that's because each one is going to give us maybe slightly different information. We have some that gives us information about deeper pressure, some about vibration, some about fine touch. Because we have so many of these different types of receptors sprinkled throughout, we're able to receive a lot of sensory information. So let's see here. Here's a free nerve ending here. Sits up a little bit higher. This one, it says tactile corpuscle. It's the same thing as messenger corpuscle. Here's a Merkel cell, sensory nerves that they're branching onto, and root hair plexus, and then a Pacinian corpuscle. So they're scattered all throughout. Okay, tickle and itch. We have receptors that are specific for the sensations tickle and itch. And these are interesting and they're clinically significant because they're pretty easy to stimulate. And a clinician could actually map out if you have any sensory nerve damage based on if you're able to receive like tickle or itch information. So that's one way that they can be used. For tickle, these are also free nerve endings. It's usually described as pleasurable, but there's a psychological factor there. Like for me, for example, if you were to tickle me, I would say that you were torturing me because I absolutely super hate being tickled. And then itch is interesting. They're probably the same nerve endings as tickle. You do have specific itch spots, but there is a potential for itch to be considered even more unpleasant than pain. And I usually have a few people in each class that when I say that, they kind of go, oh yeah, which for me, I'm not one of these people. Like if I feel itchy, then I itch. But for some people, that itch sensation is so strong, and it's even stronger and more uncomfortable than pain. Yes?
Yeah, I'm just a little confused because I definitely know there are tickled spots. There are spots where the sensation of tickling is more intense, but I don't really. I haven't really think of any spots as being specific itch spots.
Oh, specific itch spots. This really means more of there's areas where we don't have receptors for itching. Yeah. And I know that we can feel itchiness like all throughout our skin, but that's pretty much superficial, so any deeper layers aren't going to have any itch spots. Okay, baroreceptors. Baroreceptors give us information about changes in pressure. So these are stretch receptors. So again, when your bladder is full, the reason why we can feel the bladder being full is because the stretch receptors, the baroreceptors in the bladder, are being pressed against from all the urine that's being held in the bladder. These are also free nerve endings, and any organ that has the ability to stretch is going to have these baroreceptors. Organs that have the ability to stretch are called distensible organs. If you've heard the term distended, sometimes that pops up in like medical drama, TV and movies, like, oh, is the abdomen is distended. It just means that it's stretched out, it's puffed out. These are also very fast adapting. These baroreceptors are very, very important. These help regulate internal functions. So when baroreceptors in the bladder are triggered, we know to go to the bathroom. When baroreceptors in the bowel are triggered, we know we have to go sit on the toilet. When baroreceptors in the heart or the lungs are triggered, that is going to change our breathing rate or our heart rate or blood pressure, which can have really large changes on the body. This is why when you're checking someone's pulse, the best spot or the first choice spot to do it is on the radial pulse, which is on top of the radius, which is on the thumb side. You can check a person's pulse in other areas. You can check it on the femoral artery, and you can also check it on the neck, which is the carotid artery, which is very large. But by checking the pulse on the carotid artery, what you are in fact doing is placing a little bit of pressure on the carotid artery, right? Well, those carotid arteries are super important because the carotid arteries are sending blood up to the brain. You really need that blood up to the brain. So if you are going to have pressure placed on the carotid artery, baroreceptors in the carotid artery say, oh, we've got a little bit of pressure pushing against us. Well, we're a critical artery, so we're going to increase pressure in the blood to overcome the pressure being placed against us. So you could actually increase someone's blood pressure by checking carotid pulse. Now, if you're checking it very quickly and then you take the fingers away, there's probably no harm in that. But if you're dealing with somebody who has high blood pressure issue, you really don't want to do anything that's going to further increase the blood pressure. Proprioceptors give us information about where our body is as far as position, our muscles, and our joints. We have 3 different types of proprioceptors that work together that give us that information. We're actually going to take a look at how muscle spindles and Golgi tendon organs work when we do our next physiology lab in lab. But 3 different types are muscle spindles. These sit in the muscle. The muscle spindles give us information about changes in the muscle length. So these can trigger a stretch reflex, and that's the lab that we'll do. So you've probably all had this happen when you're at the doctor at some point. You're dangling off the edge of a table and your leg's dangling there, and the doctor comes up and whacks your knee with a hammer. Yeah? You guys all know what I'm talking about? That is a stretch reflex. We are going to look at that a whole bunch in lab. When that happens, that hammer comes in and taps on the tendon. So that triggers the Golgi tendon organs. The Golgi tendon organs monitor changes in tension on the tendon. So the tendon suddenly has this high tension placed on it. The tendon is attached to the muscle. So the muscle gets pulled on all of a sudden. That changes the muscle length. The muscle spindles send that information, and we end up having a reflex because of that. Question or comment?
Yeah, just was wondering if the Golgi tendon organs had some relation to the Golgi apparatus themselves, or is that just they were discovered by the same guy?
Golgi tendon organs and Golgi apparatus. I don't think so. Obviously, it's the same name, but no. And then joint capsule receptors sit within the joints and give us information about pressure within the joints.
Okay, it's just the same guy.
And then I think this is my last one. Chemoreceptors give us information about chemical changes in the body. Really important ones, as an example, would be our blood pH and levels of CO2. These are things that we are not consciously aware of. So this information goes up to the brainstem, and is controlled automatically. We do not have to consciously process that information, but it is extremely important in order to keep our pH at its happy homeostatic level and our CO2 levels as well. Okay, I think we're doing pretty good on time. All right, questions on this piece here? Okay, I'm going to pull up what we didn't finish from last time. All right. And you're going to see this in lab this week too, so that's a nice overlap there. So in this lecture, what we were doing was just going through and naming the different parts of the brain and what they do. So we had just talked about the diencephalon, which is here. And now we're starting with the midbrain. The midbrain is the first piece of what is called the brainstem. The brainstem is made up of the midbrain, the pons, and the medulla oblongata. This is actually a really great picture to reference when you're looking for your terms this week in lab. So the midbrain here sits just below the diencephalon. The midbrain processes visual and auditory information and then produces reflexes from that information. So it's not terribly bright in here right now, but if we were to step outside and it was really, really sunny, that would cause our pupils to react and constrict to not let too much light in. That reaction occurs at the midbrain. because we are reacting to that visual information. No. Okay. Pons. Pons is the next part of the brainstem. And we can see here that the pons is actually connected to the cerebellum. So it's the connection point. Here we have both somatic and visceral motor information is being sent to the pons and then being controlled by the pons. So these would also be automatic things that we don't have to consciously control. Breathing is a good example, and we talked about this before, because we are able to consciously control our breathing, but we don't have to, which is why we can breathe when we sleep. So that type of motor control would be directed in part by the pons. And then we have the medulla oblongata. The medulla oblongata is the last part of the brainstem, right before the spinal cord starts. We also have relayed sensory information here and very important functions like heart and breathing rate. So in the brainstem area, we see a lot of automatic functions, breathing, heart rate, reflexes, which is why the brainstem is super critical and why damage to the brainstem could be catastrophic. Then we have the cerebellum. The cerebellum is sometimes referred to as the mini brain. It sits just inferior to the cerebrum. And this programs a lot of involuntary motor movements, like your postural muscles that's keeping you upright is controlled in the cerebellum. And then we have connections from the cerebellum to the brainstem and the rest of the spinal cord. Okay, the peripheral nervous system. So everything before that, the central nervous system was the brain and the spinal cord. The peripheral nervous system is how the rest of the nervous system is connected to the central nervous system. Part of the peripheral nervous system are the cranial nerves. We have 12 cranial nerves that are very special because they are connected directly to the brain here. They sit on the inferior part of the brain. So this picture here is if you were to take the brain and look at the underside of it, you would be looking at the cranial nerves here. I will go through the cranial nerves with you on the models in lab very specifically so you know where they are, but we will go through them together and talk about them here. This is a great picture for you to reference when you are going through the terms in lab and need to find the cranial nerves. I will not ask for location and I will not show you a picture of the cranial nerves for this class, but I will ask questions about the name and what the cranial nerve does. So these are the first 6. These would make good flashcards. And then I'll tell you a few different mnemonics that you can use to help remember the cranial nerves and what they do. So the first 6 here. Olfactory is number 1. Optic, ocular motor, trochlear, trigeminal, and abducens. This is one of those things that a lot of repetition is a good thing. Olfactory, optic, ocular motor, trochlear, trigeminal, abducens. Let's take a look at what these nerves do. So olfactory, the first one, you might already be familiar with the term olfactory. This helps with our sensation of smell. Optic is, we have the optic nerves that actually make a cross. This gives us visual information from the eyes. Ocular motor, so the ocular part, what's that? Yeah, very good. So the motor part tells us that something's moving. The ocular part tells us that something's going on with the eyes. So it controls the eye muscles and shape and size of the pupil. So when the pupils either dilate or constrict because of light, it's controlled by oculomotor. Trochlear, I don't have a tricky way to remember this one, but that controls one eye muscle. Trigeminal receives sensory information from the head and sends motor information to the chewing muscles. So this is a mixed nerve that does both sensory and motor. Abducens controls a different eye muscle. Okay, the second half, I know it's a lot, but repetition, and we'll see it a lot in lab too, so it'll start to become more familiar. Facial, vestibulocochlear, glossopharyngeal, vagus, accessory, and hypoglossal. So these are listed in order. So facial, I feel like that name kind of helps because facial makes you think of the face. This helps control facial expression. And then what else is on the face? We have our eyes, so it helps control tears, salivation, and taste information.
It can be
Vestibulocochlear. Have you heard of a cochlear implant? A cochlear implant is something that goes in the ear to help someone with hearing. So vestibulocochlear, the cochlear part, that can help you remember that is responsible for hearing. Glossopharyngeal. So pharyngeal, we've had that name pop up a few times. This is in reference to the pharynx, which is an area of the throat. So it controls areas of the pharynx, also controls the tongue, and responsible for taste.
Okay, so is the difference between the glossopharyngeal and the hypoglossal? The hypoglossal is like moving your tongue for talking and such, and the glossopharyngeal is for moving your tongue for like swallowing.
Yeah, good. Vegas, not spelled like Las Vegas, but it makes me think of Las Vegas. And when I think of Las Vegas, I just think of like all the bright flashing lights and all the noise, and there's just like a bunch of information coming in from everywhere. So that helps me remember that it receives sensory information from lots of places. So we have the tongue, the diaphragm, the esophagus, and lots of different areas. So the vagus nerve receives lots of sensory information. Accessory controls a muscle in the neck and the back. And hypoglossal, again we have that word glossal, voluntary muscles of the tongue. So the best way to tackle these cranial nerves is to remember them in order. Because there is an order, and it's the order that I just presented to you. It's 1 through 12. And this mnemonic can help you remember the nerves in order. So old opie, and the first few letters of each word in the mnemonic match up to the first few letters of each cranial nerve. So OL for old gives us OL for olfactory. OP for opie gives us OP for optic. So old opie occasionally tries trigonometry and feels very gloomy, vague, and hungry. There are a lot of mnemonics out there. If this one doesn't really click with you, you can look up other mnemonics. I have a different one that I had a student tell me my first year teaching, and it's stuck in my head, but it's really dirty. So if you want me to tell you, I'll just ask me and I'll tell you. Or you can make up your own. I've had students make up their own, whatever works for you. But it really does help because the words are kind of long and they're very new. So the mnemonic helps to remember in order. And then the second mnemonic down here will help you remember the function. So in this one down here, S is for sensory, M is for motor, and B is for both. So some say merry money, but my brother says bad business merry money. So for example, the first one, olfactory, so old. And then, oh gosh, what does olfactory do? Okay, some say, Some, S is for some, it's sensory, and that can help you remember, or maybe just the word olfactory can help you remember that it's smell information. So on the exam, there will be questions about, I'll either say, like, the olfactory nerve gives us information about, and then the A, B, C, D, or I'll do it the other way, where it's which of the following nerves controls an eye muscle, and you'll select from those. There will be a handful of questions like that, enough to where it's definitely worth committing these to memory, because you're going to see these in lab as well. No, great information, right? Good news. Okay, almost done. A nerve plexus is just a cluster of nerves that branches off from the spinal cord. So the spinal cord, the whole way down the spinal cord, we have all of these branches. But we're going to have a lot more branches that come out that innervate basically the limbs, the arms and the legs. So we have these clusters of nerves called a plexus. The cervical plexus we can see up here in the cervical region is this cluster here. The nerves that branch out from the cervical plexus are going to innervate areas of the neck, of course, because it's right next to the neck, and the diaphragm, our muscle involved in breathing. The brachial plexus, brachial might make you think of brachii, brachialis, makes you think of arm. So the brachial plexus, that branch or that cluster, are going to innervate the shoulder and arm. We don't have a plexus until we get to the lumbar region. Lumbar plexus innervates areas of the pelvic girdle and the upper thigh, and the sacral plexus, this cluster down here, innervates down the leg. Question?
Yeah, don't we have plexus somewhere in the chest area?
I think solar plexus. Okay, yeah, that's not. It's not the same brand. Yeah. Okay, for the autonomic nervous system, we can break this down into two categories. We have the sympathetic and the parasympathetic division. You might be familiar with these by their nicknames. So the sympathetic division is our fight or flight mode. This is the nervous system mode that we kick into in emergencies. So adrenaline is released. We are ready to fight or run away. We are in emergency mode. Versus the parasympathetic division is rest and digest. Rest and digest is hopefully what everyone is experiencing right now, where your body is functioning at normal operating levels. Your digestive system is working normally, your breathing patterns are at a resting level, your heart rate, your blood pressure is at a resting level. If a tiger were to suddenly come in here, we would all very quickly jump into our sympathetic division, where all of a sudden we have energy that was stored is going to be released. Where do I have that? Stored energy is released. So we're going to release glucose. That glucose is going to go to the skeletal muscles, so that way we are primed and ready to either try to outrun the hungry tiger or fight off the hungry tiger. Also, during the sympathetic division, all non-essential systems are shut down because it is emergency mode. So certain systems are going to take precedence over others. Our cardiovascular system, which is going to be our heart pumping, that becomes critically important. Things like our digestive system, those get shut down. We don't need to digest the hamburger if the hungry tiger is going to eat us. So blood is shunted away from non-critical systems and delivered to basically the skeletal muscle and the cardiovascular system. I think this is my last one. Yeah, the last thing we'll look at is just some specific examples of the differences between the systems, between the sympathetic and parasympathetic divisions. So for our eye, during fight or flight, the eye dilates. So when the eye dilates, we're able to receive more light. So we are looking around and we are noticing things. We're getting more information. We see the hungry tiger. We're aware of movement over here. We see the hungry tiger's friend. Versus rest and digest, the pupil constricts. For our heart, during fight or flight, heart rate increases and blood pressure increases also. The idea here is get that blood pumping, get it to the working muscles. Heart rate during rest and digest is going to be decreased. For the airways, the diameter of the airways increases. So the air that we're breathing in, we're able to get more oxygen in to the heart to pump it to the working muscles. And then we're breathing faster as well versus rest and digest, where we're breathing slower. Digestion. Again, we do not have to worry about digesting that hamburger. If the tiger's going to eat us, we don't have any use for that hamburger. So the digestive system temporarily shuts down. This is why when you are feeling stressed, really stressed, if your body dips into fight or flight mode, it can mess with your digestive system. Have you ever felt so stressed that you can't eat? Or just you're having a really upset stomach and you're having to run to the bathroom a lot? The digestive system is not working in the same way when you are stressed out and in the sympathetic nervous system. When you are not stressed and you're in your happy rest and digest, the digestive system is working properly. Liver. The liver is where we store glucose in the form of glycogen. Well, we're going to need that glucose back if we're going to fight or run from the tiger, so we start breaking down that glycogen during the sympathetic nervous system. But at rest, we're going to create that glycogen and keep on storing it. The reproductive system, no time for that if we are going to fight a tiger. We are not worried about the next generation if we're trying to outrun a hungry tiger. So reproductive system, shut off. It's working fine during rest and digest. This is also why there can be reproductive system difficulties when a person's very stressed, because the reproductive system does not work at its optimal level or at all when you're very stressed. This last one here is just interesting. We don't actually cover this in 370, but during ejaculation, that actually dips into the sympathetic nervous system just for that one point. And then for the rest of the arousal phase, the erection is actually in the parasympathetic nervous system. That's something that we'll look at more in the in 371, but it's just interesting to look at now when we compare the different systems and how they work. The differences between the divisions of the autonomic nervous system are really interesting. There's a great book called Why Zebras Don't Get Stomach Ulcers. It's written by Dr. Robert Sapolsky, who's a professor up at Stanford, and it goes into basically how we as humans have this whole other level of complexity when it comes to stress compared to a zebra that has this simple life. You know, the zebra's out on the grassland eating. Hungry lion comes up, and the zebra dips into sympathetic nervous system to outrun the lion. And then it's either going to outrun the lion and be fine, or it's going to be eaten. Either way, it's only going to be in its sympathetic nervous system for a short amount of time. That's what the sympathetic nervous system is designed for a short amount of time. But humans, college students, we experience stressors that do not last just a short amount of time, right? We experience stress in much longer intervals. We've got finals weeks. We've got rent to do. We're trying to figure out how to pay for gas for the first time. Things like that, right? Relationships. So we have the potential to dip into the sympathetic nervous system much more frequently and for a lot longer amount of time compared to these zebras. And the effects that type of stress has on our body, I mean, you can see just a few examples here, but there's a lot more that happens when we live in that fight or flight mode. So if that's something that interests you, this book does a really great job at looking at all the different things that happens, and it's super interesting to me. So we can chat about it sometime, but I know that you guys that was a lot. So I'll leave it there for now, and we can chat more about it if you have any questions. But anything for now? Okay, so study guide posted later today, review on Thursday.